Hi, my name is Sophia Williams, and today I'm going to be talking about biological safety cabinets and how to use them and what they're used for. So to start off, a biological safety cabinet is pretty much to create as sterile of an environment as possible. And they can also be called laminar flow hoods, and this is because the flow that they create is laminar and it is non-turbulent. And the flow hoods that we use in our lab are vertical flow hoods, and it could be vertical or horizontal. And it is very important to know the flow of the hood. That way you know how to use your hands and how to operate inside of the hood to make everything sterile. So since ours is a vertical flow, the flow flows from the top to the bottom and sort of back. And so what that essentially means is that anytime you put something over a sterile object, that object is no longer sterile because the air above it is pushing all the sterile air down, and so it will get the non-sterile air from the object and be pushing it down into the sterile environment. Here we have our simulated biological safety cabinet, and I gathered all of my materials. So what I have are going to be some Kim wipes. This is the liquid that we're going to be moving around and stuff. I have my laptop for my protocol and some ethanol in a spray bottle, and then I have some like simulated beakers that I would be mixing things around in, and then I have straws to simulate pipettes. And here, when you first look at a biological safety cabinet, you have to ensure that these two displays have a green light above them. So these are all going to be a ton of buttons, and there's going to be an on button that you press and a light button. So you're going to turn it on and turn the light on, but you can't begin working until you have these two green indicators. And so once you turn it on, you're going to have a clear covering right here, and you're going to have to pull the cover up until it reaches this little hash marks. So the first thing that I do before doing anything in the hood is I take my ethanol with in the spray bottle, and I spray my hands with gloves on them. I just kind of rub my hands really quickly, and then I would spray the ethanol all over the surface of the biological safety cabinet, and then take a Kim wipe and wipe the area entirely down. That way there's nothing left over from previous experiments. And then before we can actually start putting things into the cabinet, we have to spray everything entirely down. So we are assuming that all of these packages come in sterile packages, but only the inside of them is sterile, so we're trying to just disinfect the outside but not really have a sterile exterior. So we're assuming all of these cups and things that I'm putting in here are in sterile packages. So you spray the outside, then rub it in, and then you put it in the hood. And then I'm gonna spray this one, rub it in. Next, we're going to spray our pipettes, and this is what we're going to be using to be moving liquids around. And these are also going to be in sterile packages, but again, we are assuming the outside is not sterile. And then, so now that we have our items in the cabinet, I'm going to be showing you how to pipette some things back and forth while maintaining a sterile environment. So I'm going to grab a pipette tip or pipette, and I already poured some water into this container, so we're going to be working with these two containers for now. And yeah, so since they're both open, that means we have to be very, very careful not to put our hands above them, otherwise they won't be sterile anymore. So I'm going to try and show you to the best of my abilities. Since these aren't actual pipettes, it's going to be a little bit difficult, but I'm going to show you how that would work. So here we're going to be trying our best not to put our hand in it, and then we're going to be moving that to the next one. And sometimes I like to tip it a little bit too to help with that. And that's how you try and get it to be as sterile as possible. Like I said, it is a little bit more difficult because I'm not working with an actual pipette, but you have to try your best not to get your hand over it. Now I'm gonna show you something that would make it not sterile. So if I were to just do this, for example, and just grab the water like that, you can see for both of them, I would have gotten this one not sterile by putting my hand over it, and then I would have transferred that and gotten this one not sterile as well, since it's directly above. So that's pretty much how to transfer liquids in the BSC and make everything sterile. Another thing that you want to keep in mind is the layout of things. So I try to have certain things laid out in a way that's easy for me to not put my hand over them. And also opening caps. So you want to, if we were to open this cap, you would want to try and open it with one hand, and then be careful with the cap. And grab it really carefully like that. And when working with the caps, you want to be sure if you're going to put it down or anything like that, that you put the cap face up. That way it's only getting sterile air and it's not touching the surface of the BSC. So that is all for this. And once you are done running your experiment, you would just trash everything, and you would spray the BSC with ethanol and a Kim wipe like we did in the beginning. And if you were to take your wastebasket out and dump your pipette tips, you would have to respray the basket and put it back in. So this is pretty much how you would work in a sterile environment inside one of these cabinets.